Thank you very much for your kind introductions and very I have to say thank you for the organizing committee to let me share my experience in the neoantigen identification in osteosarcoma. This is my first talk on this topic, so I'm very excited to share you our recent result. Um, so let me briefly you about osteosarcoma, that why we've been working on this type of cancer for like 10 years. It's because it's a bone cancer that is very high incident in children and adolescents. And also the survival rate of the patients with metastatics or the patient with not respond well to the standard treatment that include chemotherapy plus resections has a very low survival rate in 5 years. And this one is actually not very improved from 30 years. It's improved because of chemotherapy, but after the introductions of chemotherapy, everything is still the same for 30 years ago. So we try to find a novel treatment, novel therapy for osteosarcoma patients. So that's why our group would like to focus on the thing. This is a neoantigens identification. So let's move back to the genomic landscape of osteosarcoma because it's a rare cancer, it's pediatric cancer. This slide gonna show you that the tumor burden or mutational burden is very low comparing to the adult cancer. Osteosarcoma is here. So this figure is not very promising that you're gonna say that, oh, it's very heavily low mutation burden, so why we gonna look for the neoantigens? Because normally the tumor burden is very positive correlation with the neoantigens. But I have to say that the genomic landscape of osteosarcoma is very unique. It's not about the point mutations or SNV or indels, but it's about the structural variations. In most of the case, it has the rearrangement of the genomic landscape that resulting from the chromotipsis, that is the fragment of the DNA shuffle that combine in different way, inversion, or combine it differently. So you're gonna see that there are so many structure that change in osteosarcoma patients. And also they have a cluster of mutations called cartilages that is different in the structural variation at specific areas. However, very few recurrent clinical actionable alterations have been identified also in osteosarcoma. So that means that we really need a personalized medicine for each patient. So I would like to say that structural variation is actually can create many types of neoantigens. This picture is actually shows that once you get the structural variations, you're gonna see the rearrangement of genomes that include maybe a single genes alterations or the whole chromosome changes from the amplified deletions or relocalizations. And this can create novel open reading frame. That means you're gonna get plenty of neoantigen from the structural variations. This graphic actually shows that the frame shift mutation is actually occurred because of the open reading frame that has been shifted. And also the deletion and duplications also give rise to the structural variation derived neoantigens. It also occur in 2 genes and 1 genes. I wanted to show this. It's very interesting because this study is actually identify structural variation derived neoantigen from up to 2,000 tumors. And you can see that osteosarcoma is here. Comparing to adult cancer, osteosarcoma is one of the most highest neoantigen derived from structural variations. So it's very promising that we're gonna look at the neoantigen that derive from SV. So we firstly started with the 2 genes combinations. So we can call it fusion genes. The fusion genes can be different types, including in-frame fusion, frame shift fusion, or out-of-frame fusions that include the coding sequence and non-coding sequence. And after this, you're going to have so plenty of neoantigens that might be expressed in osteosarcoma cells. So let's move to the fusion neoantigen in various types of cancer. This is a neoantigen score for the fusion genes versus SNV or indel mutations. You're going to see that the neoantigen score of the fusion genes-derived neoantigen is very high compared to the SNV or indel mutations. And also when we calculate in the neoantigen permutations, it means that in one mutations in the fusion gene can generate many neoantigens comparing to the SNV or indels. So let's move to our project. We actually granted by the Genomic Thailand. We really have a chance to do multi-omic analysis, whole exome sequencing, RNA sequencing, and also proteomic analysis in 100 osteosarcoma tissues in 3 years. However, this work is actually just in the pilot cohort. So we started with 21 patients that we have the tumor tissues before the treatment and after the treatment. And we send the tissue sample to perform RNA sequencing, and then we do the neoantigen identifications. Before we identify the neoantigen, we do the fusion gene analysis, and we predict the neoantigen from the fusion genes. We actually very stringent filter the neoantigens according to the high MSC class 1 binding affinity, high properties of MSC processing and presentations, and also the high T cell recognition ability. This one actually worked by Dr. Sukulat and Dr. Chaiyawat. Let me share you our results. In each column is actually from each patient. We have 21 patients. This sample is from the pre-treatment samples. It's very striking that we found that 100% of the patients carry at least one fusion genes. And the one fusion gene can lead to the more number of neoantigens. So you're going to see a positive correlations of the number of neoantigens and number of fusion genes. And this number is actually compared with the previous study that they can identify fusion genes in pediatric cancer patients. So we try to profile that the difference between each group of the patient according to the aggressiveness of the disease. So we classify patients into stage 2B and stage 3. Stage 2B is a patient with localized disease, and stage 3 is a patient with lung metastasis. The blue graph shows the number of fusion of patients with stage 2B, and the green shows the patients with stage 3. We actually haven't seen any significant difference in the number of fusion genes between these two groups. And also, we haven't seen any significant difference between the number of neoantigens between these two groups as well. So it means that all of the patients carry the same number of neoantigens from the fusion genes. And also the patient with good prognosis and poor prognosis. Patient with good prognosis means that after getting chemotherapy and surgical resection, they haven't developed any recurrence or lung metastasis within 2 years, let's say, or they can live longer for like 5 years. And the poor prognosis patients always develop lung metastasis or recurrence within 2 years after diagnosis. These two groups are also the same. We haven't seen any significant difference in number of fusion genes and neoantigens. So that means that we might be able to identify fusion genes from all of the patients at very same levels in our cohort. So let's move to the pre and post treatment tissue because this one is actually the past sample from the same patients. We got the pre-treatment tissue at the diagnosis, and after chemotherapy and resection, we got the post-treatment tissue. For the number of fusion, we found that in some of the cases have the higher level of fusion genes. And also the same in the neoantigen numbers, we found that some of the patients have the higher number of neoantigens. So it's very close to what we expect because we expected that after the chemotherapy treatments, some of the mutation is increased because of the chemotherapy. Yes. And this one is actually the list of the peptides that is a neoantigen peptide from fusion genes that we identified from these 5 patients. And you can see that some of the patients have the share neoantigens, but not that much, only a few. But in one patient, we cannot see any share neoantigens pre and post chemotherapy. Some of the patients had very high share neoantigen, maybe because they already have the high number of neoantigens at the beginning. And also together, we found that 50% of the neoantigen identified are not shared between pre and post chemotherapy. So that means that if we would like to identify the neoantigen to give rise to the vaccine approach, maybe we need two at the time that we would like to give that therapy because the mutations are changed all the time. Tumor evolution often occur at the time. And this one go to the type of the structural variation that we can identify. It's actually so many type of SV that we can identify, including deletion, duplication, inversions, or translocations. The blue graph is the post-treatment and the red graph is the pre-treatment. This graph shows number of neoantigen per mutations. That means that one fusion gene that we identify can generate how many of the neoantigens. And we found that the type of mutation that gives the highest neoantigen is the inversions in pre-treatment specimen. However, for the post-treatment specimen, we found that the duplication mutation generates highest neoantigens. So I would like to give you an example of the list of the fusion gene from the inversion mutations in pre and post-treatment. You can see that there are no shared neoantigen in this mutation type. But the thing that I would like to highlight is that the fusion gene that we can identify is from the coding and folding. So the in-frame or out-frame mutations, and also the coding and non-coding regions joined together. This one is almost my last slide, actually. So as I mentioned, that we actually did a very stringent filter of the neoantigen identification that I have shown you. So this one, we use the algorithm to predict MSC binding affinity, and we found that most of the neoantigen that we can identify has a very high MSC binding affinity, as well as the T cell recognitions. We also found that neoantigen that we identify had a very high T cell recognition because the T cell recognition is much more 0.5%, and the AFC binding is less than 500 nanomicromolar. So last but not least, my talk today is, I think fusion gene is a new class of neoantigens that very promising candidate target for the treatment of osteosarcoma. And I have to say that is actually not very specific to osteosarcoma because there are now so many type of cancer that have the fusion genes, and structural variations is one of the most prevalent mutation they found in cancer. And so we try to develop this pipeline to use the platform to not only use for osteosarcoma, but for another type of cancer that have the very same patterns in genomic landscape, just like osteosarcoma or other type. So I would like to explain my deep gratitude to express our respect and thanks to the patients and their parents for their participation in this study. This slide is very important because we have very good collaborator. Dr. Nunsai is actually not our collaborator, he is our boss as well. Thank you for his effort to collect all the tissue sample for 10, 20, 15 years. 15 years. And all of the patients or the tissue that we collect, we have the very good clinical data, so we can match it very perfectly, and we can see a lot of things in this cohort. And Ajahn Jongwani, who is a pathologist, expertise in bone and soft tissue sarcoma. And one of the important team is the biobank team. They did a very hard work, help us to collect all of the tissue samples, blood, and everything. So we have a lot of specimen in our bank right now, waiting for the grant to do more multi-omic analysis. And also the bioinformatic team, data and statistic team. And also I would like to thank you the grant, the Genomic Thailand, SORSO, and actually PMUB. That is the very good program to upscale our researcher in the team to have the skill of bioinformatics in order to identify all of the neoantigens. I have to say that we are trying to develop new pipeline actually and new algorithm to precisely predict neoantigen. Maybe we're gonna focus on every type of neoantigens, but however, right now we still have only one sequencing data available. We're waiting for whole exome sequencing data that will be coming out in maybe in this year. So maybe we're gonna have a lot more to share you in the next conference. And also thank you for my team, my Salmon Concert research team in CMU team. Thank you.
Thank you, ladies. You already promised that you're gonna give a talk next conference. So, if you any question from the floor, ครับผมเชิญครับ
Thank you for very interesting research here. I have a question for metastatic patients. If is that possible to expand your cohort to work with the metastatic osteosarcoma? Because just to see that if the immune system or antigenicity in the metastatic site remain the same as primary tumor. Because in my experience in the medical oncology field, even though the tumor contain high immunogenicity, for example, high MSI high or a defect MMR, or actually high TILs when we send the patients to tissue to test for comprehensive genome profile or RNA or TILs. When we give immunotherapy, for example, checkpoint inhibitor, the patient never get any response. So it doesn't mean they were not going to get any benefit, but as far as I know, that there's very low antigenicity, and I'm not sure about neoantigen in the metastatic site. Also, if we see a lot of literatures now, is to combine between immunotherapy and targeted therapy to close this gap and to get any synergistic effects between response between checkpoint inhibitor and targeted therapy without any reason. I mean, some synergistic mechanism. Thank you.
Thank you very much for your discussions. We actually trying right now to expand our cohort to the lung metastasis. That is the major site of this hematic in osteosarcoma. But the thing is that how to get the tissue sample for lung metastasis a little bit different. It's a little bit harder than the primary tissue because he's not a surgeon. Other clinic surgeons that cover with this one, but right now we are trying to collect that tissue. We have some in our biobank right now, and actually some of the paraffin embed might be another thing that we're gonna use. Is the samples may not be the great quality, but maybe we can try. We can try it as well. Yep.
Do you have any problem with decalcification of the bone tissue? I mean, it's difficult because the bone cancer too.
Maybe he would like to answer these questions.
Not really.
Yeah.
The osteosarcoma is a lack of the bone
Mm.
actually. So
Yeah.
we can get a pure tissue from the metastasis or even in the tumor site.
Yeah, nice works. Thank you so much for joining us, and we hope to, see more of your work later. So, what is the response rate of osteosarcoma to checkpoint inhibitors?
It's not really.
Yes.
Yes.
I don't think it's not response.
I think it's very minimal.
Yeah.
I can say
Yeah.
it's 1%
Right.
Yeah. Sorry about that. But okay.
Answer.
So that's mean you see this quite high neoantigen from fusion genes, but in the reality,
Yeah.
it doesn't really work, right?
Yeah.
So I think one thing is maybe you need to do some immunological work as well. So if you can get some blood PBMC and test it,
Yeah.
whether you can see that. So to make sure that okay, it can induce immune response, but with the tumor microenvironment that suppress it so checkpoint is not working.
Yeah.
So are there any comment from Ajahn Pairat?
Yeah, I think since you have the those genomics data, I would be interested to see all those variants in the whole pathway, right? Beta 2 microglobulin,
Yeah.
HLA themselves. I mean, you can look at it. And also look at the HLA expression, MSC expression. I think that I'm not sure whether it would be a combination of those.
Yeah. We actually have the data. We haven't shown it today, but according to the tumor microenvironment in osteosarcoma, we know that it's an immunosuppressive, and so it's very common in the low tumor burden cancer. But we found that actually the fusion genes, neoantigens, has a very high immunogenicity, so it's work very well with the setting of type of cancer that had a low mutation burden and low immune infiltrations as well. So we think that it might be another promising. And also for the validations of our result in wet lab, we are planning to do it. Maybe we're gonna collaborate with Ajahn Chiang for this, because right now we have only the bioinformatic tools and data that available. So we are actually position us as the target identification persons. So we really need to you to help us to do more work.
So you use the pipeline that you develop in-house?
Right now we use the pipeline that available, but with some modifications. Yeah. But our team gonna use their skill in order to find new algorithms or use the machine learning in order to find the match peptide with the MSC. That's a very critical point. We did a lot work on this, so I hope that we can have the very good pipeline after this. Because we have data right now.
So I have one question from online.
Yeah, so there is a question from online. So it's come just from the beginning of your talk. The anonymous attendees asked, does the epigenetic is this possibility to be used to find the neoantigen of osteosarcoma?
Oh, you mean methylation patterns or acetylation patterns? I have to say for osteosarcoma, we haven't have the information about the epigenetic changes in osteosarcoma, but in other type of cancer, maybe. Yeah.